Hello everyone, my name is Caroline and I'm a final year PhD student at Imperial College in London. In the next 5 minutes, I will introduce my PhD project, which is aimed at developing a novel screening platform for highly multiplexed biomarker analysis. But why do we need methods for multiplex biomarker detection? Well, one of the reasons is that we are all very different. We all express diseases differently, we have different genes, and we all live very different lives. And because we express diseases differently, we need new and better methods to detect these differences in order to make the most accurate diagnoses. Ideally, we want the method to be more sensitive than current technologies, because the more sensitive the technology is, so the lower the detection limit, the sooner we can find biomarkers in the blood and therefore are able to diagnose diseases earlier. We also want the method to be highly multiplexed so that we can look at a large number of molecules simultaneously. And finally, we want to achieve these results as fast as possible. So ideally, we want a method that achieves high throughput in order to save time. The ultimate goal is to develop a highly individual multiplex disease detection platform, which moves us closer towards personalized medicine. To achieve this, we have developed a new detection strategy which uses the ONT sequencing platform in combination with newly engineered barcoded molecular probes. The probe consists of two main parts. First, the barcode sequencing region, which is a 35-base sequence, and second, the target binding region, which is either a complementary microRNA sequence or an aptamer. Since the barcode sequence can be easily changed, a large number of barcoded molecular probes can be designed, which serves as the basis to enable sequencing and identification of a large number of molecules. To determine the presence of a molecule, we differentiate between two states, no target versus with target. Since the target binding region can be easily changed, our method allows us to look at microRNAs, proteins, and small molecules. If no target is present, the barcoded probe translocates through the pore, and the DNA barcode is base called, which determines the identity of the probe. If, however, the target is present, the signal of the electrical current changes. This is due to the unzipping of the double strand or the unraveling of the aptamer. A delay in the signal can be observed, and this delay can then be quantified using different delay algorithms. So far, we were able to perform multiplex biomarker detection by using 3 different barcoded molecular probes. All 3 have a different barcode, and the target binding region binds selectively to either microRNA 208, thrombin, or serotonin. If we look at the percentage delay, we see a significant increase when targets are added compared to the control where no targets were present. To the best of our knowledge, this is the first method achieving simultaneous detection of several biomarkers. Moreover, we were able to use the same technology looking directly at human serum of 8 participants. The goal was to find the microRNA signature, so kind of like a fingerprint, for each participant. We found that some of the 40 microRNAs we looked at were significantly more present compared to the control, where no serum was present, suggesting that we were able to detect those microRNAs in serum. In summary, I've presented to you our strategy which employs barcoded molecular probes in combination with nanopore sequencing. Furthermore, I have demonstrated the feasibility of detecting multiple biomarkers simultaneously. Finally, I have showcased our ability to directly detect 40 microRNAs in serum. As I conclude, I would like to express my gratitude towards my supervisors Joshua Ede and Alex Ivanov, as well as my postdoc Ben. Moreover, I want to thank ONT who have partly funded this research, as well as the other funding bodies EPSRC, ERC, and the CDT from Imperial College. If you want to read our latest publication and follow me on social media, please scan this QR code and reach out to me. Thank you.